Dr. Minton. I'm here in the Eye Research Institute at Oakland University. This is our shared tissue culture facility, cell culture facility. These are called Biosafety Level 2 or BSL-2 cabinets. They allow you to do work safely with any infectious agents inside of this hood space. You'll see these on the news all the time where lab technicians are working in them. There's HEPA filtered air from the ceiling of the cabinet, and it pulls air in away from the operator. And this is what we use to do a study where we are testing volunteers who may have had and recovered from COVID-19 to see if they have any antibodies for a certain time after their infection. So this is called a lateral flow assay. This is one that we just ran on a volunteer. It has one control line, which all the tests have if they work properly, but no other lines, which means that that person does not have a high level of antibody to the COVID-19 in their blood, which actually would be expected from the length of time they've had the infection, which is quite a few many months ago. We use this particular brand of tests from Epigenetech that we've been testing because I know for years they've made very good specific reagents. And these lateral flow assays were problematic around March and even January, as early as January, where companies were just making tests that didn't work and had false positives. But this test has no false positives, and so we're doing this study that we really started doing a lot of tests about August. So these are examples of tests that have been UV sterilized now, but just kept for examples. Here's another negative test, but here's two tests where the volunteers have an extra line, and that's telling me that they have IgG antibody to the COVID-19 protein, which they were expected to probably have in the test group that they were in. And so that's one thing we were able to do here and start this project at a time when regular research was stopped really in the state of Michigan. But I'm a biochemist even though I do vision research, and this was something we could do with the help of some of our med students to get this protocol going and do tests in the community here in Oakland County and surrounding counties. So that's what a lateral flow assay looks like. Very simple. It develops in 15 minutes, and if the test is one that's reliable, it can be deployed to test for antibodies and needs no special equipment really. We use the biosafety cabinet for the time being. When we started this study, applying to do the study, we really didn't know about the degree of infectivity of COVID-19 and wanted to be very precautious even with a finger drop of blood. Thank you. Have a good day.